our body consists of hundreds of different cell types, and yet they all come from a single fertilized egg cell, the zygote, which in a couple of divisions in the uterus of the mother diversifies into what's called germ layers. These are three different major types of primitive cell types. And each of these germ layers generates a subset of cells, like mesoderm makes blood and makes muscle, ectoderm makes neurons and skin, etc. And so this is how the different cell types are generated, but we really understand only very partially about how this diversification works. And one way to study it is to use a model system which sort of mimics what's happening in the animal. And these are the famous embryonic stem cells that are capable of making all three germ layers in culture, and where we can do mechanistic studies. And what people found in previous work is that the transition from these very primitive cells to the three different germ layers happens in two discrete stages. First, the embryonic stem cells sort of forget what they are, and then they acquire a new genetic program, which could be either mesoderm or ectoderm or any other of the germ layers. And so that it looked like these were completely separate processes, and a number of regulators were found that affect one or the other. In our study, very surprisingly, we found that a single regulator, whose name is WHSC1 because it's related to, in its knockout form, to a genetic defect in humans. And this gene, when we destroy it in embryonic stem cells, makes that the cells get stuck in the primitive state, and it also affects the way they differentiate. When we then induce these cells to differentiate, they will only form one germ layer out of the three. And so we have the very surprising discovery that there is a new regulator that links what we call pluripotency exit with differentiation on the one hand, and it also has a high specificity for only two of the germ layers. And this gene is also interesting because a very closely related gene has an important role in a certain type of cancer, the acute myeloid leukemia. And as I mentioned, in a rare genetic disease, it causes developmental malformations. So this is the main message.